thousands of unidentified human remains are discovered around the world. Some victims are found days after death, while others remain hidden for decades. As time passes, the body decomposes, fingerprints disappear, and facial recognition becomes impossible. Yet, one silent witness often remains: DNA. But what happens when even DNA has begun to degrade? Can science still uncover the truth?
Hello and good morning everyone. We are Group 24 and today we will be presenting a review of the article Analysis of Human Degraded DNA in Forensic Genetics, which was published in 2025. The main purpose of our presentation is to show you how the scientists identify individuals by using degraded DNA. Imagine that investigators discover skeletal remains buried in a forest after more than 30 years. There are no fingerprints, no identification card, no facial features left to be seen. The only remaining biological evidence is DNA. But after so many years of exposure to the environment, can the DNA still be able to identify the individual? That is the question. And today we will be answering that question. DNA or deoxyribonucleic acid is often described as the blueprint of life because it contains the genetic information that makes every single individual unique, except for identical twins. In forensic science, DNA profiling has become one of the most reliable methods for identifying unknown individuals. It plays a very crucial role in criminal investigation, missing people cases, victim identification, and even archaeological studies. This is because every person's DNA is unique, therefore it gives a strong evidence that can link the biological sample to a specific individual. However, the DNA is not indestructible. Once a person dies, the natural repair mechanism inside the cell will stop functioning, and the DNA will start to break down. Naturally occurring enzymes will break the DNA strands apart. Water causes hydrolysis, which removes the DNA bases, while oxidation damages both the DNA bases and the sugar backbone. At the same time, bacteria and microorganisms invade the body tissues and release enzymes that accelerate the DNA degradation. Environmental factors such as high temperature, humidity, UV radiation, and soil conditions further increase the rate of DNA damage. Despite these challenges, advances in forensic genetics have made it possible to recover useful genetic information from highly degraded DNA. This review explored the latest technologies that allow forensic scientists to identify individuals even when the traditional DNA analysis fail. Understanding these advances is very important because it helps to solve cold cases, identify disaster victims, and reunite families with missing loved ones, and also to bring justice through scientific evidence.
Thank you, Shad. As we have learned, DNA begins to degrade immediately after death. However, the rate of radiation depends greatly on the environment in which the body is found. According to this review, several environmental factors influence DNA preservation, including temperature, humidity, soil chemistry, ultraviolet radiation, microbial activity, and burial condition. Temperature is considered one of the most important factors. In hot environment, chemical reactions occur much faster, causing DNA strands to break down rapidly. This is why DNA recovered from tropical climate is often more degraded than DNA preserved in cold regions. Humidity also accelerates DNA degradation because moisture promotes the growth of bacteria and fungi. These microorganisms release enzymes that continue destroying DNA after death. Soil conditions are equally important. Acidic soil and certain minerals may damage DNA, while ultraviolet radiation from sunlight create chemical change that make DNA even more difficult to analyze. Interestingly, not all body tissue preserve DNA equally well. Soft tissue decompose rapidly because they are easily attacked by microorganisms. In contrast, bone and teeth provide natural protection for DNA. Their dense structure shield genetic material from environmental damage, allowing DNA to survive much longer. For this reason, forensic scientists often collect samples from bones and teeth when investigating unidentified human remains or archaeological skeletons. Once suitable sample had been collected, forensic scientists begin the DNA extraction process. Because degraded DNA is usually present in very small amounts, specialized extraction methods are required. The review highlights the use of silica-based extraction technique. This method binds DNA molecule while removing contamination such as soil particles, humic acid, and other substances that could interfere with laboratory analysis. After extraction, scientists measure the DNA concentration using DNA quantification techniques. This step is extremely important because it determines how much DNA is available, whether inhibitors are present, and how severely the sample has degraded. This result helps scientists decide which analytical method, STR analysis or SNFP analysis, will produce the most reliable result. Another critical aspect of forensic DNA analysis is contamination control. Even a tiny amount of DNA from investigator, laboratory staff, or the surrounding environment can contaminate the evidence and produce misleading results. Therefore, forensic laboratory follow strict contamination prevention procedures, including wearing protection clothing, sterilizing equipment, and maintaining clean laboratory environment. Now that we understand how degraded DNA is preserved, recovered, and prepared for analysis, the next question is, how do forensic scientists actually identify a person using this damaged DNA? To answer that, I would like to invite Ika, who will explain the different DNA analysis techniques, including STR analysis, SNLP markers, and the revolutionary technology known as next generation sequencing.
Well, thank you, Rafiza. After DNA has been successfully extracted and purified, the next step is to analyze it. The choice of DNA analysis method depends on the quality and quantity of the recovered DNA. So let's begin with the short tandem repeats, commonly known as STR. STR are short DNA sequences that repeat several times at a specific location in the genome. The number of repeats varies from one individual to another, making STR highly useful for human identification. However, STR analysis has one major limitation. It requires relatively long DNA fragments. When DNA is highly degraded, many of these fragments are broken into pieces that are too short to amplify successfully. To overcome this limitation from STR, forensic scientists increasingly use single nucleotide polymorphism, which is called as SNP. It involves a variation at a single position in the DNA sequence, although each SNP contains less individual information than an STR marker. It requires much more shorter DNA fragments for analysis. This makes them particularly useful when working with the degraded DNA and to recover from old skeletal remains, disaster victims, or archaeological samples. So, to compare SPR and SNP, scientists usually analyze a large number of SNP markers together. When combined, they provide highly reliable information for identifying individuals, establishing family relationships, and supporting forensic investigation. So let's go for one of the most significant advances highlighted in this review is next generation sequencing, or commonly referred to as NGS. Unlike the traditional methods that analyze only few DNA markers at a time, NGS can sequence hundreds or even thousands of genetic markers simultaneously. This technology requires only a very small amount of DNA and performs exceptionally well with the degraded samples, making it is a powerful tool in modern forensic genetics. Mm, Another advantage of NGS is that it can analyze different types of genetic markers in a single test, including SDR, SNP, and mitochondrial DNA. In addition, in addition to identifying unknown individuals, NGS can also assist in kinship analysis. In some cases, for predicting externally visible characteristics such as eye or hair color. Um, these capabilities make NGS one of the most promising technology for future forensic investigation. So that's all from me. Thank you.
In conclusion, this review demonstrates that degraded DNA is no longer any possible challenge in forensic science. Although environmental conditions can severely damage DNA, modern extraction methods have improved laboratory techniques and advanced technologies such as SNP and SQ have significantly increased the ability of forensic scientists to identify individuals from degraded biological samples. These advances have transformed criminal investigation, disaster victim identification, and missing person cases, providing a new opportunities to solve cases that were once considered impossible. As forensic genetics continue to evolve, combining scientific innovation with ethical responsibility will be essential to ensure that the DNA evidence remain accurate, reliable, and trustworthy. We hope this presentation have give you a better understanding of the importance of degraded DNA analysis and its contribution to modern. However, we also identify some limitations. Since this is a review article, it does not include any original experimental data or laboratory findings. This discussion would have been more stronger if it included more real forensic case study demonstrating how these techniques have been applied in actual investigation. In addition, the article provides a limited discussion about the financial costs or practical challenges of implementing advanced sequencing technologies in routine forensic laboratories. After reviewing this article, our groups have identified several strengths. First, this review provides a comprehensive explanation of how DNA degrades and why analyzing degraded DNA is challenging. Second, it clearly compares traditional and modern forensic technique, making it easy to understand the advantages and limitations of each method. Finally, the article is supported by recent scientific reference and discusses emerging technologies such as next generation sequencing, which are shaping the future of forensic genetics. Besides technical challenge, forensic genetics also raise important ethical concerns. Modern sequencing technologies can reveal much more than a person's identity. They may provide information about ancestry, family relationships, or even certain physical characteristics. While information can be valuable in criminal investigation, it also raises concerns about privacy, informed consent, and the potential of misused genetic data. Therefore, forensic scientists must follow strict legal and ethical guidelines to ensure that genetic information is used responsibly and only for legitimate forensic purposes. Although modern forensic DNA technologies have greatly improved, the analysis of degraded samples are not without challenges. The main challenges of degraded DNA is that only a very small quantities are often available. This increases the risk of incomplete DNA profiles and may reduce the accuracy of the identification. Another challenge is contamination. During evidence collection or processing, DNA from investigated or surrounding environment can accidentally mix in the sample. Even a very small foreign of DNA may affect the reliability of the result. In addition, advanced technologies such as next generation sequencing require expensive equipment, specialized laboratories, and highly trained personnel. The large amount of sequenced DNA produced must also be analyzed using sophisticated bioinformatics software, which increase both the complexity and the cost of forensic investigation.